Thank you so much, everybody, for coming and being at this meeting. So my name is Diana Barucha-Goebel. I'm a neuromuscular neurologist at Children's National in DC, as well as at the NIH. And so it's a pleasure to be here today to be able to talk to you and introduce this session. We have a great lineup of speakers. Let's see. Oh, is it possible to just keep the slides on? There we go. Okay. Thank you. So you'll be hearing from a bunch of people about updates in the CMT4. So hopefully everybody's in the right room. And if you need, so you'll be hearing from Dr. Stephen Gray from UT Southwestern, Dr. Cleopas Cleopa from Cyprus, Dr. Alessandra Bolina from Italy, Dr. Riccardo Perfetti from CMO Applied Therapeutics, and also from Terry Pira Velasquez from Elpida Therapeutics. So we have a really great lineup, and to give some updates on what's been going on and what are some future updates as well in this field. So briefly to introduce the CMT4s, and again, I know I'm talking to some folks who themselves are the experts and are living through this or know someone or are doing research in this field. So bear with me, but basically just, when we're talking about the different CMTs, we're talking about at least 100 or more different genes that are causing these, the hereditary neuropathies as a whole. And there's ongoing gene discovery work and ongoing genes that are being identified, and still, again, a subgroup that remain uncharacterized from a genetic perspective still. So there's still work being done and more work to do. These are a diverse group of genes and really involve various pathways that are linked to the health of the nerve cells, the axons, Schwann cell health that then feeds into myelin integrity, et cetera. And so not to get too much into the weeds here, but with the CMT type 4s, we're talking typically about demyelinating neuropathies that are phenotypically heterogeneous, meaning diverse. They can present in different ways, different age when they start or present, some with a more pure neuropathy and sometimes with some other features, central nervous system involvement or cataracts or vocal cord involvement. So there can be, some variety as well between the genetic types or even between individuals. And so I've highlighted here, while it's a very complex figure, just the ones underlined in red are the genes that we know of, at least right now, that are linked to the CMT type 4s. And again, maybe a few months from now, that list might even look different because there's really, this continues to grow. And so with the work with the CMT Association and through CMT Star, and you were hearing already a little bit about this in Catherine's talk that in the general session, the CMT Association is really working to try to target CMT from many different perspectives. And this is in addition to things you heard about this morning around how to optimize function, activities of daily living and care for CMT. But this is really taking it a step further to say, how can we be ready for advances in therapeutics? And so this goes to various genetic targeted therapies, additional drug development, understanding our biomarkers and being ready for clinical trials, and really partnering academically as well as with companies in advancing this technology. Sorry for that bottom right is cut off with the video. But there's also, again, work that we've talked about in gene discovery. In addition to that, there's also various efforts underway in natural history study. Maybe some of you are part of that as a participant or as a clinician or researcher. And this all helps us to really feed into being ready for clinical trials and making it, again, allowing for that launching pad for the therapeutic development and to really be ready to bring these into the clinic. And then in addition, even both within the CMT field, but also even outside of the CMT field, advances in drug development, and especially around neuromuscular and neurogenetic, especially when you talk about genetic therapeutics, there are advances happening in parallel in this field that are going to continue to feed back as well into the development in CMT. So it's a really exciting time, and there's a lot going on. While there are many genetic causes of CMT, and we're talking about the CMT4 subtype here, the advances really, are in common really to various subtypes of CMT. So thinking about, gene therapy targeting, the diagnostics, natural history work, and also drugs that may be available or can be developed to improve the health or stability of the nerve axons as well. And so there are gene therapy efforts underway for various subtypes of CMT that are in different stages of development. You'll be hearing about some of them today, so I don't want to take up too much time here. But I will briefly just talk a little bit about what are we talking about when we're talking about genetic therapeutics. And again, we're going to be diving a little bit deeper into that during the session. You can think about different kind of boxes or categories. So gene addition or gene replacement, correcting not at the DNA level, but the messenger RNA, or also things like gene editing, or inactivation. And so these span from various phases, from preclinical to clinical trials to some products that are also approved in these different categories. So with gene replacement therapy, what we're talking about is using a virus, or you'll see it also referred to as vector. Typically, we're using AAVs or adeno-associated viruses. These are delivered by various routes depending on where you're trying to target. It could be delivered into the bloodstream or into the spinal fluid. And these get into the cells. They then get further into the nucleus of the cells. The coat or the envelope, it uncoats, it disintegrates, and you have ultimately the actual DNA that sits as a circular material or episome within the nucleus and has the ability to continue to produce protein in the body, hopefully in those target cells where it is deficient. So for CMT specifically, then you can think about what are the routes. So how would we want to target the different subtypes of CMT? Most likely we'd be talking about routes where you go into the spinal fluid space, so intrathecally, so through the spinal tap type procedure, or intravenously might be options that could be explored and are being explored. And then you can think about, well, what kind of virus are you going to use? And so there are different AAV subtypes or strains or serotypes, if you will, that have a better proclivity towards targeting certain cell populations in the body. And so you might see those, again, in the drug development, and especially AAV9, which you'll be hearing more about, especially from others today in this talks, is being used quite commonly for CMT research, both to better target the peripheral nerves and also the goal of trying to target our Schwann cells as well. So there's various initiatives in the gene therapy space. Again, I don't want to talk too much about these because you will be hearing about some of these in the session, but around some of our different CMT4 subtypes. There's also a project, exciting work by Dr. Bolino on the CMT4B1 that you'll be hearing about, and also some exciting advances as well in the SORD clinical trials that you'll be hearing as well. And so again, there's ongoing work and support in affiliation as well with the CMT Association, looking at especially around some of the gene targeting and gene therapies, as well as natural history work, as you can see here. So just to highlight a few, CMT4C, 4A, 4J, and also 4B1 and B2. So I don't want to dive too much into those, although it's a little bit of a wordy slide, but you'll be hearing about these updates directly from some of these experts who are doing this work today. So with that, I will pause, and then we'll go ahead and hand over to our next speaker. So I think we were going to move our schedule around and have Dr. Perfetti next, who's from Applied Therapeutics. So thank you.
Thank you. So I will cover some of the science and clinical development activities around the SORD deficiency. Patients with SORD deficiency have a defect of the pathway. Is the pathway that metabolizes glucose into a utilization source that is very poorly used by many species. Glucose has many routes of utilization. The primary one is to produce energy. We also have pathways to store glucose in case we need additional energy. There is one very secondary pathway, which is called polyol pathway, where about 3% of glucose that we manage by making it or absorbing it gets routed through. So it has a very secondary implication. Does not produce energy. Is probably an evolutionary remnant of a purpose for this gene that in species starting from fruit fly to human is not really serving any critical purpose, but the pathway is active. In patients with SORD deficiency, because the inhibition of the second enzyme in this pathway, the sorbitol dehydrogenase, they're not able to make fructose. That is not the major problem because fructose could be synthesized ex novo by the human body, but also we absorb quite a little of fructose through what we eat. The problem is primarily that by having that defect, they accumulate the step before. So the step before is sorbitol. So patients with SORD deficiency have an excess sorbitol. That excess of sorbitol is toxic in many different ways, and that is responsible for the disease. Here you have some of the background information on the disease. I mentioned a little bit about the mechanism. We don't have currently therapies for SORD deficiency, but we have been working at blocking the step prior to the making of sorbitol, the one that from glucose lead to sorbitol, so that we could offset the excess of sorbitol from a different directional approach. The science around SORD deficiency from the first publication, Dr. Mike Shai was one of the authors of the 2020 Nature Genetics publication, to the phase 3 clinical trial, which started in 2022, is a bit unusual. I mean, I've been in science and clinical development for quite a long time. I think 2-year time from the discovery, or at least the public knowledge of the discovery, and the phase 3 is probably unmatched. But the journey was actually very long, from in vitro, cell data, animal models, first pilot study to the phase 3. I will tell you some of the information that may be relevant. The panel that you see on the left of the screen shows the level of sorbitol that the cells collected from patients with SORD deficiency produce. The gray bar. You see that there is an excess of sorbitol of cells that we collect from patients. Even when you put them into a petri dish, they still manifest the phenotype that they have in humans. If you give them an inhibitor of the making of sorbitol, an inhibitor of aldose reductase, which is called 8007, we are able to significantly decrease the production of sorbitol. The panels on the right part of the screen demonstrate how in an animal model you could demonstrate that indeed you are reducing sorbitol, and that translates into benefit. So the animal model that was used is a Drosophila. In science, is one of the animals we use the most because the life cycle of Drosophila is very fast. So you don't necessarily need to wait for years to develop a phenotype. The upper left panel, you see this very elegant structure that is stained by green fluorescence. Even if you're not a scientist, you can tell that the panels on the left part of the screen are very harmonic, very elegant, very structured. You see in the middle, the pointer, doesn't really work, I guess. But you see the panel in the center where that structure is disrupted. So that animal has been engineered to carry the SORD gene deficiency. if you expose the animal to the inhibitors of aldose reductase, you are able to rescue that structure. And we have many different tests to measure how the animals moves in space. Animals that carry the gene defect have a clear phenotype. They move slowly. They don't have a sense of direction. And the treatment with the study drug shows a rescue of the phenotype. Stefan Zuckner has also worked at a rat model that also recapitulates a similar phenotype. What you see here is the first pilot study we conducted in 2021-22 on with patients with SORD deficiency. So what is measured here is the level of sorbitol. The gray bars are the level of sorbitol of patients before they entered the study. And the green bar is the level of sorbitol that they had after exposure to the study drug. So there was an important decline of the sorbitol level, but the study was also relevant because we wanted to make sure the drug was safe first, what type of administration we needed, once daily was sufficient, and also to see whether the drug had a sustained effect over time. Sometimes when you block an enzyme, you have an overriding. The biology try to overcome your intervention. No, this was stable and permanent. What was very important to note is the level of sorbitol of patient at entry correlated with the severity of the disease. So all of this science fed into the development of the phase 3 study, which is currently ongoing. We will have very soon the first evaluation because the last patient that entered the study will reach month 12, and that's where a set of assessment will take place. So I think that this could be my last slide. Thank you for your attention. We're taking questions as if there is any.
Yeah, since Dr. Breakout has to leave, if anyone has a question right now, let's go ahead and open the floor, please.
So what's the target patient population for this disorder?
The one defined by the genetics. So for the study.
We're talking numbers. Do you have a
So in the United States, there is an estimate of less than 4,000 patients who may be affected by the disease. In Europe is slightly larger. Characterization of other regions of the world is still poor. So the publications that have been out are in those, typical Northern Hemisphere part. But those are the numbers. Yes.
Ricardo?
With regard to that last question and some of the newer genetic approaches, which we talked about earlier, do you think those numbers might increase?
Significantly, right? So the genetics, which in the publication Dr. Shai was author in 2020, identify a primary gene defect with one specific mutation. But then science, as often evolves, there have been recent identification of other mutations that may actually significantly change the numbers. So it remains a rare disease, but the numbers I provide you may need to be corrected up.
So are you also doing any work on sort of a companion diagnostic aspect that goes along with it besides just the improvements in genetic testing?
We have developed the assay to measure sorbitol in blood, and we have validated the assay from an FDA standard point of view. So obviously, we may be able to partner with companies that actually are, laboratory companies that could do that. Yeah. Thank you.
Thank you, Dr.
Thank you.
All right. So our next speaker is a pre-recorded talk from Dr. Cleopa. He was unable to make it to the meeting in person, and so we have a video talk from him that will be projected.
Good afternoon. My name is Cleobas Cleoba. I would like to welcome you to this session during the CMT Patient and Research Summit, and I would like to give you a brief update on our work on CMT4C gene therapy. CMT4C is a relatively severe and early onset form of demyelinating CMT neuropathy. It shows faster progression compared to other common CMT1 types and results from mutations that affect the SH3-TC2 gene. It is among the rare autosomal recessive CMT neuropathies the most common and makes approximately 3 to 4% of all non-CMT1A CMT neuropathies. So here we have a loss of function mechanism, and the disease is caused by the loss of function of the SH3-TC2 protein, which is an important molecule within the Schwann cells that form myelin in the peripheral nerves. It's a part of the recycling compartment of vesicles within the cell. It is crucial for the proper development of the cell and also for the formation of myelin, and mutations have been shown to disrupt this function. So based on this knowledge about the cause of CMT4C, our aim is to replace the healthy gene in myelinating Schwann cells. In order to achieve that, we developed a gene replacement therapy that is specific for Schwann cells. First, we had to design a therapeutic vector that includes the human SH3-TC2 gene, and we had to place that under control of a myelin-specific promoter element, the minimal version of the myelin protein 0 promoter. We packaged this therapeutic DNA into adeno-associated viral vector and delivered it to the mouse model of CMT4C by lumbar intrathecal injection. This is a clinically relevant delivery approach that can be applied also in humans. We showed that we can achieve adequate biodistribution in the peripheral nervous system of these mice, and this leads to the expression of the normal SH3-TC2 protein in the cells of interest in myelinating Schwann cells, and in fact, in a high percentage of myelinating Schwann cells. Using this technology, we then proceeded to perform treatment trial in a CMT4C model, which is deficient for this SH3-TC2 protein. We treated these animals both at early as well as at late stages of their disease, and evaluated the outcome by testing their motor performance and also by evaluating the nerve conduction studies. So we showed that these mice benefit from the treatment. They show improved mobility of the legs. You can see here, there is improvement in the performance on a rotating rod. There is improvement in the grip strength, and there is also improvement in the nerve conduction velocities and in the magnitude of the response. We also looked at the morphology in the peripheral nerves and demonstrated that we have improvement in myelin formation. We get thicker myelin sheaths and fewer fibers that are completely devoid of myelin sheath. We also looked at a particular aspect of this disease, which is the elongation of the node of Ranvier. This is an area of the nerve that is very important for transmitting signals. It is abnormal in CMT4C patients and in the mouse model. And so we showed that we can actually correct this abnormality by treating with the therapeutic vector, and we bring the length of the nodes of Ranvier to the same range as the normal mice. So we are making progress on the way towards developing treatment for CMT4C. We have until now achieved proof of concept that we can have a therapeutic benefit in the model of the disease by replacing the SH3-TC2 gene. We also performed a subsequent dose response study that I don't have time to show that confirms the initial study. So the further pathway should include the testing in larger animals. This is important for demonstrating the scale-up potential and safety. And also we need natural history studies because these are not available for CMT4C, and this will enable, at the end, the performance of successful clinical trial. So for the large animal testing, of course, we need support from industry, which is something we're working on. And for natural history study, we also need support both in terms of funding and patient participation. So we recently, in collaboration with Professor David Herman in Rochester, submitted a proposal for a natural history study for CMT4C. The aim is to enable the enrollment of 60 patients in major referral centers, and these patients will be evaluated in detail with sensitive clinical outcome measures, with biomarkers including the MRI of the legs and blood biomarkers, in order to understand the course of the disease over a period of 2 years. And this will be crucial for leading into future successful clinical trials. In addition, we want to establish a global registry to include all patients that we can reach. This is very important because this is a rare disease. We need to know how many patients are out there, what is the stage of the disease, even if we don't have detailed outcome measures in these patients. So both of these efforts are going to facilitate support by industry and the successful planning of clinical trials for CMT4C. So I would like to thank you for joining and for your attention. I would like to thank the members of my team for doing this work, our collaborators for the contribution, CMT Association for funding this work. I would like to thank the CMT4C Patient Task Force. They have been the driving force behind progress over the last few years. They also do all the fundraising efforts. This is a recent cycle for CMT event we had in Boston, which was very inspiring. And I would like to thank you again for your attention.
So that was an amazing talk. But I guess we'll be moving on since it's not here in person, but we'll be moving on to our next speaker is Dr. Stephen Gray from UT Southwestern. And you'll be hearing some more updates on some preclinical work.
Yeah, so just, again, I'm coming here from Dallas, Texas, at UT Southwestern Medical Center. And all of my work focuses on developing gene therapy treatments for neurological diseases. And so I work on a number of different diseases, but that includes a few forms of Charcot-Marie-Tooth disease. Let's see. And that's just an image of a part of the spinal cord. Those bright green splotches, they're all motor neurons where we've delivered a gene and expressed it in motor neurons. So when we talk about gene therapy, just to bring it down to the basics, right, is all of you are aware that you go to the hospital with some kind of condition or some kind of symptoms, and then through a diagnostic odyssey, you might get diagnosed that the cause of that disease is from a gene defect. So conceptually, gene therapy is going to be conceptually simple, that we're just going to take a working copy of that gene, package it in some kind of carrier like a virus, and put it back into the body. And so this would be like gene replacement therapy. That's again, conceptually kind of simple and straightforward, as opposed to like gene editing is where it's going to try to deliver sort of molecular tools to change the DNA in your body, like to kind of correct the DNA in the body. Whereas this is just literally like taking a working copy and putting it on top of the bad copy. So you're not really changing the bad gene in the body. You're just giving a functional gene on top of it. So, but the tool to deliver genes to the nervous system, I mean, there's a lot of different ways of delivering genes. One would be like the COVID vaccines. But most of the different tools that we have to deliver genes into the body are not permanent. So you have to administer them over and over and over again. This is like AAV is kind of like a molecular mail truck that's been developed as an engineered virus to deliver genes. And the idea here is if you're in a cell like a neuron, then the gene delivery is permanent. So it's one injection, and then the gene is there permanently. So that's the advantage of gene therapy, of doing it this way. But if something goes wrong, you can't stop taking the pill. So that's one of the things is you're developing like gene therapies, you have to be really, really careful that you're getting it right the first time. And it is like, we call it like one and done, but it's also, since we're using an engineered virus to deliver the gene, we actually do a really good job of immunizing patients against that virus. So basically can't get the therapy again. So again, it's trying to make sure that you get everything right the first time. And this isn't, you can think about it more as a molecular mail truck now than a virus. So it's not a virus in the sense like you're not going to deliver this and all of a sudden it's going to replicate and spread and cause a zombie apocalypse. Okay. This is, it's literally just like a vehicle for delivering genes. Now there's been hundreds of clinical trials for different diseases now using AAV, and there's 5 FDA approved products that are actually marketed and sold for things like spinal muscular atrophy, hemophilia, Duchenne muscular dystrophy. But these also come with very high price tags. I think that the spinal muscular atrophy drug is about $2.1 million for that one dose. Okay, so going directly, thinking about the nervous system, this is just a graphic of work that we have done and published, and lots of other groups have done similar work of taking this AAV delivery vehicle, injecting it into the spinal fluid by like a spinal tap. And these are just images from mice. The ones on the top are of the brain. All the dark there is where the gene that we delivered is expressed permanently. The bottom images there are sort of cross sections of the spinal cord, and those kind of dark splotchy cells, kind of in the. It's hard to describe without a pointer, but yeah. All right. Anyway, those are motor neurons, and then but it's also the sensory neurons we can deliver to those very efficiently as well. So we can treat the spinal cord really, really well with this approach. I think when we get into the brain, it's okay, but it's definitely not perfect. So this is just a kind of a short list of diseases that just my lab works on. There's a couple dozen diseases that are in clinical trials right now using this same general approach. And we've been successful moving, we've got 7 different disorders that are in clinical trials now using this approach. Giant axonal neuropathy kind of falls under the CMT umbrella. It's a very rare form of CMT. And Diana is going to talk about that a little bit later because we started a clinical trial in collaboration with the NIH in 2015. So now we're, 8 years out. And 14 patients that were treated in that trial. We're also developing, we're kind of advanced stage of development for a gene therapy approach for CMT4J that we're now doing that. We were doing that we continue to be doing that with the Talia Duff Foundation, and now Elpida Therapeutics has gotten involved with that. That you'll hear about later. And then it's kind of a newer project where we think that this could apply for CMT4A. And the idea here is to think about this as like a platform approach. We know how we can deliver the genes. We've developed protocols to do this in humans, so we can put different genes into the virus and use this as sort of a platform approach. But the trick is finding the diseases that kind of fit. So, we're delivering to neurons which are going to project the axons. We're only delivering somewhat to the Schwann cells. So it depends on like the CMT type if the Schwann cells are really the main cell that's being affected, this may not be good enough. I think we're still trying to figure that out. But if it's a type of CMT where it's the neurons themselves that project the axons, then I think that those fit into this delivery paradigm really well. So in terms of kind of streamlining the development of gene therapies, and thinking about this as a platform approach, is we kind of have the design aspect of it that we've got a lot of experience with. And we're trying to make this as like open access and sort of playbook type approach as we can for the rest of the field, is to have kind of set designs that we can use. And then it moves into like a formal toxicology study, efficacy studies in cell models or animal models, and then the manufacturing of it. And all of that is sort of templated in terms of how you would do that. And then moving into a clinical trial design that, again, we're in terms of how we're monitoring safety, managing immune responses, all of these things is pretty standardized. But the trick here is, well, one, it has to work. You know, we have to pick the right CMTs to apply to this. But I think the bigger challenge is the manufacturing component is the big beast in this in terms of sucking up cost, because these are very expensive technologies, and the manufacturing alone can run $2 to $4 million. And that's just to make enough to support an early clinical trial. So I think I'll stop there. And if we have questions about gene therapy and gene editing at the end, then I'll be with the panel discussion. Thanks.
Great. Thank you so much, Steve. Our next talk is also a recorded talk. It's from Dr. Bellino, who couldn't be here today. So we have that also as a video.
Good evening. My name is Alessandra Bolino. I am a professor at Vita Salute San Raffaele University in Milan, and I'm also working in the Ospedale San Raffaele in Milan. Thank you very much for your kind invitation. This first slide summarizes what was and is our contribution to CMT neuropathies, in particular in the CMT4 group. As you can see here, we focus our attention on CMT4B1 and B2 in principle that are due to the loss of myotuber-related protein 2 and myotuber-related protein 13 genes. We identified those genes, and then over the years, we generated animal and cellular models for these neuropathies, in particular CMT4B1. We investigated the pathomechanism behind MTMR2 loss in particular, and then uses these advantages to design therapeutical strategies to test efficacy for CMT4B neuropathies. We also, over the years, investigated a bit another form, that is CMT4J, that is due to the loss of this protein, FIG4. We generated as well animal models for this neuropathy, in particular mouse models with the loss of this protein specifically in Schwann cells. And by doing this, we also found that these two genes and proteins, so this one and this one, are functionally connected in Schwann cells. We focused our attention particularly on CMT4B1 over the years. This is a severe autosomal recessive demyelinating neuropathy with childhood onset that is caused, as I said, by mutations in MTMR2. This MTMR2 is a phosphatase, so it's an enzyme that removes a phosphate residue from lipids in this case and not proteins. And is characterized into the nerve pathologically by the presence of these myelin outfoldings that are these degenerative figures of the myelin that is here that surround normally an axon, as you can see here, the myelin in black. And then you have these extra overloops of myelin here, in this case more than one, that then tends to degenerate, causing axonal problems, loss of fibers, and then ultimately problem to the nerve physiology and, of course, muscle physiology. Over the years, we generated, as I said, several animals and cellular models for this neuropathy in particular. The aim of our CMTA-funded project was to assess whether niaspan which is a drug based on niacin-nicotinic acid, could be effective for the treatment of CMT4B1 caused by loss of MTMR2 by administering to the mouse that we generated daily this drug by oral gavage, so oral administration, daily for 5 consecutive months. Then another aim of this project is the natural history studies on CMT4B1 and B2, which means understand how the clinical phenotypes in patients evolve over the years. And this is also useful to identify specific outcome measures to monitor the efficacy eventually of therapeutic strategies. This aim is ongoing and is done in collaboration with Davide Parison in Milan, that is coordinating a clinical effort that is also involving many other clinicians in UK, in France, in Europe, and particularly in US. Concerning the preclinical studies that I mentioned using this niaspam, this drug nicotinic acid, we analyzed the trial that we performed, basically doing all this analysis that you can see listed here. Target engagement means that we try to understand whether the drug efficaciously reached the target, so the nerve, and changed some parameters. That was an indication of being active where we want and how we want to be. And this was successful, in particular expression studies in the nerve that were done in collaboration with Professor John Svaren. Then neurophysiological measurements to measure, for example, nerve conduction velocity that in CMT4B1 and in the mouse model is severely reduced. And the treatment with the drug improved the nerve function. This was okay as well. We also tested the neurofilament levels in the blood of these mice, where neurofilament are a marker of axonal and nerve degeneration, and this was decreased following drug treatment, so suggesting an improvement somehow. And concerning morphological analysis of the nerve, we found that the drug was able to reduce the number of degenerated fibers, but surprisingly not that much the myelin outfolding, so the figures that I showed you before that are hallmark of this neuropathy. In the meantime, we worked to a proposed mechanism for CMT4B1 over the years that is based on the following scheme. So we arrived to the conclusion that MTMR2 and MTMR13, which is the other gene responsible for CMT4B2, interact in a complex involving other molecules, as you see here in yellow, to downregulate the level of a lipid, in particular at the membrane into Schwann cells, and ultimately these signaling pathways that are very important for myelination. So what happens, and this is our hypothesis, is that when these proteins in CMT4B1 and B2 are lost as a consequence of genetic mutations, this lipid here, which is the substrate of action, is increased, and this leads to the increase of signaling pathways downstream, so an uncontrolled degenerative growth of the myelin in the nerve of patients and the animal models. Using this knowledge, we recently designed other 3 different therapeutic strategies based on use of inhibitors, so small molecule compounds that can interfere with this pathway. And we are in the process of testing the efficacy of these strategies still in the MTMR2 knockout mouse. But very, very recently, we decided to go on the origin, so the basic of the problem, which is the loss of MTMR2, so the enzyme that is lost on CMT4B1, as I said. So we are testing the efficacy of a gene therapy approach that is based on enzyme replacement, means replace back the enzyme that is lost due to mutations in patients and of course in the mouse, based on AAV9 viral vector, adeno-associated viral vector delivery. We think and we hope that this approach can benefit CMT4B1 and B2, as we said before, but also other neuropathies of the CMT4 type, like CMT4H, that is due to the loss of this protein here, that very interestingly is due to the presence of myelin outfoldings in the nerve, which are the same figures that I show you degenerating in CMT4B1 and B2. Thank you very much for your attention and the kind invitation. Bye.
So I'm going to be talking a little bit about our experience in an ongoing clinical trial for giant axonal neuropathy. And so this is work that's done at the NIH, or National Institutes of Health, under the Bonneman Lab with PI Dr. Karsten Bonneman, and with the support of a large clinical and lab team there. And that was also developed in collaboration with the lab from Steve Gray and his team preclinically. So briefly, giant axonal neuropathy is an autosomal recessive childhood onset disease that is neurodegenerative. It affects both the peripheral and central nervous system, and it's caused by a deficit in functional gigaxinin, the protein in this disease, that leads to basically the accumulation or dysregulation of various intermediate filaments, and they accumulate in tissues throughout the brain, spinal cord, the peripheral nerves, and you get these large or giant axons, hence the name of the disease. It starts early on with a very early sensory-driven ataxia, so unsteadiness with the gait. Also, then with progressive weakness from the motor neuropathy component. There is also the central nervous system involvement that further worsens function, especially in terms of cerebellar dysfunction and coordination difficulties. And these kids have additional systemic comorbidities with the disease, including changes in vision, bladder function, digestive issues such as constipation and GI dysmotility, as well as respiratory insufficiency and eventually respiratory failure. Some kids also have additional CNS issues, including learning difficulties and or seizures as well. So from the peripheral standpoint, giant axonal neuropathy is a length-dependent sensory motor neuropathy. So again, sharing in common a lot of the basic physiology as well in other CMT types. And in GAN, in our natural history study, we've looked at nerve conduction studies in both upper and lower extremity nerves, and many of in this room are familiar with the nerve conduction studies. And here, this is just quick plots that show the changes over time in the natural history of the disease. And so on that top graph to the top left, one of the nerves where we found a really kind of concise or clear drop is that you can see a decline in the in one of the upper extremity sensory nerve responses over time in children with GAN, and that we were able to detect that by 9 years old. We really can't in various kids, and each of those dots are different children that and adolescents to young adults we were studying. We can no longer detect that response. We see a similar pattern as well in the sural. That's one of our lower extremity or leg nerve responses or sensory responses. That also has a very clear drop, but it was really absent in many of the kids, even by 8 and onward. And then we see a similar, but maybe a little more variable, but similar drops as well in the motor responses that are the graphs to the right. And so we did explore this as well in the clinical trial. So to segue over then, in the trial, this is a phase 1 gene therapy trial that uses AAV9 with a weak but synthetic ubiquitous jet promoter. And again, this is one-time dosing. So there were 4 dose levels used in this clinical trial. And as Steve was talking about as well, we used a similar approach again with a lumbar intrathecal dosing. So it is a spinal tap procedure. And we also did have patients in a Trendelenburg sort of tilted head down position to try to increase the transduction, basically the distribution throughout regions throughout the spinal cord and towards the brain. 14 patients have been dosed, and we have data analyzed up to 6 years out, although we're collecting ongoing time points as well currently. So the children that were in the study, there were 14 kids, and they ranged in age from 6 to 14 years at the trial entry. At this age for GAN, these were kids that were already symptomatic. And on the bottom, I know it's a very busy table, but more the point being, these children already had low or even in some cases absent sensory responses. So again, that's important to think about as well in terms of then potentially as the response to treatment. And again, another point here is that there's both the disease is driven by the central and peripheral nervous system. So again, that has to also be taken into account when thinking about the response to therapy as well. So overall, in terms of the safety in the clinical trial, so we have ongoing safety data as kids continue to follow up in the study. There were one of the what we call serious adverse events, meaning a patient that required hospitalization, was a few weeks after the gene therapy for fever, vomiting, could have been another illness, but because of the time frame that was still within those first few weeks after gene therapy, we thought it could possibly be related. But overall, it's been really well tolerated. So we have not seen serious adverse events or safety issues linked to the gene therapy in our assessment. Many of the adverse event or safety issues we have identified, more than half of them have actually been related to the underlying disease itself. There are emerging class-related safety signals that are seen in the gene therapy field, especially with some of the other therapies like we talked about for SMA or Duchenne. But some of those tend to occur more when you're delivering it into the bloodstream, into the intravenously, where you can see changes in some of the heart-related enzymes, platelet and blood counts, and in the liver function. And we do not see those kinds of signals with the intrathecal delivery. So we are probably avoiding that or minimizing that by going intrathecally. We assessed efficacy overall in the trial in various measures, but I'll be reporting today mostly on 3 of them here. And one other, I skipped over, one other of the safety signals that we did see on our routine measurements. We do get spinal taps after gene transfer, and the patients did have a rise in the spinal fluid white cell count that came down on its own and didn't have any clinical signs, but it was something that we did see, and probably a response to the virus that's used for delivery. So to start with the motor findings, so on the left is a natural history plot from just the trial participants prior to their enrollment in the clinical trial. So we had been seeing some of these kids in natural history. We can detect and measure a decline in one of the measures. It's called the MFM, or Motor Function Measure Scale or score. And that is about a -7 points annual decline. The graph to the right is a little complicated, but what we looked at is, we looked at statistically a few ways, but we said, what is the probability that after these kids are getting treatment, that we are shifting that? We are either improving, and that could be anything from slowing the slope or halting the progression or improving. So what are we seeing? So those 3 of the curves that you see that are kind of to the right of that vertical line, those are 3 dose groups where we actually did see a positive, that rightward shift. Or another way that might be easier to look at as well is kind of just the raw plots. This is in long-term follow-up, the actual individual patients and how they were doing compared to the slope. So there is still some progression in a length-dependent fashion, but again, we are seeing some modification of the disease with the gene therapy. And again, these were symptomatic children, and again, with already quite involved nerve involvement from the nerve conductions as well. Going back to nerve conductions as well, so this is the natural history graph that we talked about, and a very interesting finding we've seen in now 6 of the 14 patients that were dosed is that we actually, in this case, we are seeing a reversal, that some of the kids who had absent or had already lost nerve responses to some of their sensory nerve on that nerve conduction study have a now steady increase following gene transfer, and that was actually a really exciting finding that we've seen so far. Again, this restoration here seems to occur predominantly in the sensory more than the motor nerves, and really more in the upper extremity than in the lower extremity from what we've observed so far. And then we are also looking at pathology, so from nerve biopsies. So we're doing a nerve biopsy from one of the sensory nerves at the wrist at baseline, and again, 1 year after gene transfer. It's a little bit of a complicated slide, but one interesting finding we've had is that there's an increase in something called the regenerating clusters. So these are potentially clusters of regenerating axons that might be potentially a marker here of regeneration. It's interesting because there's been, again, across patients, really just overall a positive signal or an increase in that density when you look at the density of those on the slide review. So maybe one thought is, is this, do you first have this, increase in regeneration, and then perhaps does that precede the later improvements that you then can detect by the nerve conduction studies? So overall, to conclude here, again, the gene transfer intrathecally with AAV9 for GAN appears to be safe thus far. I didn't go into it because it's a more complex topic, but we are using additional immune modulation to try to, again, dampen the immune response to the virus. There appears to be a modification in the slope by the motor function measure in this study in kids with GAN. Again, we can detect a decline in natural history in the nerve conduction studies, but the amplitudes, the size of those responses, and that we are actually seeing some improvement or even increases in some of those, especially the sensory responses in 6 of the patients. There's been this increase in regenerative cluster density that we talked about. And so kind of a point in taking all that together is that really within patients, the propensity for, to see an improvement or a stabilization may be dependent on, again, the health of those motor neurons, your dorsal root ganglia, the nerve axons, and that might differ from patient to patient, but even within one person, that may differ from nerve to nerve. So really thinking about how you're looking at different segments of the body, because you might have maybe some areas where the nerve degeneration continued and other areas that you actually have stabilized. So thinking about that as well when we're looking at studies in other CMT subtypes. I'd like to thank the team at NIH, especially PI Dr. Karsten Bonneman, a prior fellow Dr. Dima Sadeh. We also have the collaborators from UT Southwestern and at Johns Hopkins, and then also Hannah's Hope Fund and the families and patients who've been really coming and sacrificing their time to come for all their study visits. So thank you.
So my name is Terry Pervolakis. I want to thank the CMTA community for inviting me here and being part of this extended family. My journey started 5 years ago. I was an IT director for a large bank, and during my journey, my son Michael, who was born on December 17, 2017, was diagnosed on April 2nd of 2019 after an 18-month diagnostic odyssey with a disease called spastic paraplegia type 50. And SPG 50 causes low muscle tone for the first couple of years of life. Some patients have microcephaly, some patients have epilepsy, but the majority of the patients have lower body spasticity. They're paralyzed by the waist down from the age of 10 and quadriplegic by the second decade of life with severe developmental delays. And 1 to 2 months after my son was diagnosed, I attended a conference at the World Gene Cell Therapy Conference. I met 6 of the 7 world experts in gene therapy. And then I flew to Sheffield, England. I met the 7th expert in gene therapy, and Cambridge, where we were fortunate that they were studying our disease for the last 20 years. And I attended every FDA and NIH, as well as the boot camp at Ultragenics, to learn how to create a gene therapy for my son. And about a week after that conference, the ASGCT conference, I met Dr. Stephen Gray, and we partnered together to make a gene therapy for SPG50 or AP4M1. And on the right is Dr. Xin Shen, who was instrumental in helping us along this journey. And after liquidating our life savings, we realized we didn't have enough money, even if we sold our home. So we started raising as much money as we can. We did galas and barbecues and golf tournaments, and you name it, we did it. And over the 4 years, we raised $4.5 million towards the therapy that we created. And thanks to the amazing institutions and companies you see on the screen, we got Health Canada approval on December 30th to treat my son. And on March 24th of last year, my son was treated with the gene therapy that we were able to create at Sick Kids Hospital in Toronto. And let's make sure it's here. And basically, after my son was treated, we knew we had extra drug and we wanted to help more children. So we created a phase 1/2 at UTSW. We treated 2 children. One was from Wisconsin. The other child was from Spain. And then we realized that the only way to move it forward was to get approval for a drug, because the reality was that $1 million to make the drug and $500,000 to inject the children, only the rich or people that are able to raise money can treat their children, and that's gonna leave a large gap in the community. So we were very fortunate that the company that we partnered with was Virogen in Spain to make the drug, and they were able to make 12 more doses through extra drug that we had. So we're actually starting a clinical trial in Spain, And we're treating 2 children in Spain, 5 in Italy, and 5 in Germany, the entire populations within each country. And we're actually starting a pivotal study in the US in the summer. If this is a bit wrong, we have 16 natural history patients, 8 treated, and we're hoping to get approval in 3 to 5 years if we show that the drug works. So that's kind of the journey that we were on. And the reason why PETA Therapeutics came to be was that, again, we realized that no company was going to take this on with only 90 children in the world. So I quit my job. I started a company. We partnered with the NIH and a company and an institution called CERM in California. We're about to receive the funding, and we started the company. So it's me and 3 other individuals. And we took on my son's disease, that's PG50. And what's more important today is we also took on a disease called CMT4J, which Dr. Gray spoke about earlier on the science. And our strategy for this program is to take 16 to 20 patients into a natural history. That natural history is going to start in the next 3 months across 5 sites: Iowa with Dr. Shai, Stanford with Dr. Day, UTSW with Dr. Einacone, and the NIH with Dr. Bonneman. And our goal is that we collect enough information in a prospective and retrospective study that we will not have to do a placebo control or delayed start to doom our children into not getting the treatment. We will then dose 8 patients in a study in the coming year. And our goal is to find a secondary endpoint or a biomarker that will show that it's working and it's able to help the children. We are doing a phase 1 through 3. We're going to skip the phase 1 and then 2 and then 3 because the reality is after 5 years, there'll be newer technology. So we need to get it out to all the kids through insurance and through governments. And then this way, all the children and all the people should be able to get it. So the natural history study is going to start in a couple of months. We're going to do 15 to 20 control patients. There's the locations that we're doing it. We're going to try to match the treatment as much as possible. And Neurogene, who owned the program before, has a lot of excellent natural history data that we're going to add on to this as well to try to capture a really long period of time. And we're going to do retrospective digital patient records as well. So we're going to try to capture all of that to go to the FDA and say, let's do a phase 1 through 3. Right now, we're in GLP talks. We're about a month in on a 6-month rat tox study. The natural history study is going to begin. We're going to file for the IND in March if everything goes well with the tox study. And we hope to treat patients in Q4. We're going to be doing a 500-liter manufacturing run and a 1,000-liter run, which hopefully will produce 12 doses. But if not, we hope to at least get 8. I want to thank everybody that's here today, all the doctors, the scientists, and all the researchers. I should have done it like you guys are experts at that. I'm not that great at this, so I didn't have a slide with everybody to thank because there's so many people. But I want to thank Jocelyn Duff, who has been just an amazing individual and human being who started this off for her child, but continued on to make sure that all the children got this therapy. And Dr. Gray for being there whenever I needed him. And thank you very much.
Okay, so I think that's all of our lineup. So now we can go through questions for any of the panelists. Do you want everyone up, or do you just pass the microphone around?
Pass the mic, because we do have people in the Zoom room, and so you have to be on the mic so
Okay.
they can hear you. Okay, testing. Are you sure it's on? Okay, there we go.
Okay.
So I'm happy to walk around.
I'm a little ignorant to this. I've just learned of CMT last year. My girlfriend had it. And to my understanding, CMT, the research has really only evolved in the last, 10, 20 years maybe? Or is it, right? It really hasn't, from what I understand, wasn't much knowledge about it 10 years ago.
Well, I think, I mean, over time, I think you probably had advances in understanding the gene involved and the biology first. That then leads as well to the ability to understand how the disease is changing. So work in natural history, trying to identify, and this precedes even beyond 10 years ago and more, understanding, and that's maybe Dr. Shai, I can even have him speak a little bit to it, but understanding how the disease changes over time, natural history work. How do you capture change in kids or adults with CMT, right? So all of this work, it paves the way for being ready for trials. But yes, I know a lot of the therapeutic development, and it was kind of mentioned in the general session, thinking about companies that are partnering in drug development targeted to CMTs. That definitely, a lot of that movement has been more recently. I don't know if you want anything else to
Sorry, and just to follow up on that is, I'm going to remember how I phrase this, is on a timeline, I guess, to the panel here, how would you rate the advancement that has come along in the last few years, and how, with so many different CMT, 1, 2, 3, all these, do you see a treatment or even a cure sometime in the near to late future?
Yeah. I don't know if any other panelists also want to address that too. I can jump.
I'll make a general comment. Like when we talk about a cure for quote unquote CMT, it's like you're talking about a cure for cancer. CMT is like over 100 different diseases. And I think that there's a lot of commonalities where I think working together, there's an efficiency there, and there's a lot of learnings that can be shared between the different forms. But a lot of the therapeutic developments are often sort of targeted to one of those at a time. And there was the one talk from Italy where I think she's got a compound that has some general features to kind of help myelination, and maybe that could apply to more. But when we talk about like gene therapies or more targeted therapies, it's really, we're developing this therapy for this one gene. But we can use the learnings. If we develop a therapy for one, then we can use those learnings to try to transfer that more efficiently to others. So I think, unfortunately, that's kind of where we're at.
And I'll say, we've had great advances, again, both in CMT research, but even in the non-CMT neuromuscular and neurogenetic research that have even, again, led to some approved, if you're talking about even just gene therapy as an example, now some FDA-approved therapies. And with that knowledge, we understand, again, and with other trials, as we gain kind of collective knowledge about how best to target certain cells, how to measure the changes, optimizing safety, what dose to use. So, again, I think there's collective knowledge being gained that then helps kind of accelerate and we see kind of a real, pretty steep increase in the number of diseases, for example, even in the gene therapy space, where people are able to bring that to the animal studies and then bringing them to clinical trials. So I think we are seeing an acceleration in how these are being developed. And that's just gene therapy. There are also, again, other pathways that are being targeted as well, both by the CMT and in other diseases here. Yep.
So a lot of the time what happens is a researcher will have a liking towards a disease, or maybe found the disease and will work on the disease, like Cleopas has done for a few of them. Other ones are like Jocelyn and I, who have started the main research, have gotten to a certain point where it's showing that it works. And then from there, a company will take it on, or hopefully an institution, or maybe you got to take it the whole way through. So the problem really comes down to, there's so many diseases. There's 10,000 rare diseases, 400 million people affected, right? And companies are not making the money that they need to on these programs, especially when there's under 2,000 patients worldwide. So the answer, unfortunately, is if a researcher is working on that disease, it'll move forward because they're getting grants from the government. If a disease doesn't have a researcher that's really actively working on it, it'll take someone like a family foundation or a trust fund or something to that effect to get it to a certain point where it'll be appealing to someone to move it forward. That's not all the diseases, but that's why Parkinson's, ALS, Alzheimer's, the larger diseases have a lot of treatments where, a lot of the CMTs where there's 4, 5, 10 patients. CMT4J, very few, but Jocelyn funded the research. Dr. Gray made it happen and moved it forward where it took someone to move it along. Does that make sense? It's like we're in this really predicament right now where if there was thousands and they were being done, then there wouldn't be so much this. But because there's only a few of them being done, it takes a lot more, initial funding to get things going.
Over here. Yes, thanks.
Yes, we only have a minute. I'm so sorry. We do have to wrap.
I would try to be fast. And a connection, I used to be the general counsel of CERM. So thankfully you got CERM funding for that. But I think you just hit on a point that I wanted to ask you about, which is the further commercialization and distribution. So it's great that there's all of these research efforts that take place, but ultimately they need to be able to reach the patients at some point. And you hit on that key point of, it's usually for-profit companies that then have to take this on and work through the distribution. Unfortunately, that doesn't always happen. A CERM example was an ADA skid treatment that was done out of UCLA by Don Cohn. Incredibly successful clinical data. It went to a company called Orchard Therapeutics out of the UK. They had 4 assets in their portfolio. This was what they viewed the least profitable asset, and they dropped it. So it had to go back into UCLA, and the only treatment regimen for patients was to open up a clinical trial and treat this as an open clinical trial, seemingly now forever and always. So I think one thing to consider, maybe from an advocacy perspective, is getting flexibility for the FDA to look at alternative treatment opportunities. So my question, long-winded preamble here, was, what have you thought about in this situation? Because you're not looking at a robust patient population. So when ultimately you're successful and you get your BLA, what is that next step?
It's a very interesting question. So my company, just to be clear, is we're run like a nonprofit. We're not in it to make profits. So even if we had a small indication, it doesn't really matter that we wouldn't make any profits. The program that you mentioned was actually making $50 million a year. And then it was dropped. The SDA skids was actually a profitable product. And then they completely dropped it because it wasn't making enough money, which is crazy to think. Our goal is we're trying to get companies like Roche, Novartis, Pfizer to do it on a humanitarian basis, not with one program and maybe 5 or 10. Because the reality is that if they are insured and they're able to make money, but the marketing around the advocacy of taking on these ultra rare conditions, then they may take them on further. Under that, maybe the NIH, under that, if that doesn't happen, then our goal is that we will then create a commercializing entity to take them on. I don't want to do that. That's a lot of work. But if it ends up being where the product is going to sit on a shelf and never get to a child versus me having to get insurance and hire someone in every country, then we will do that. But the answer is that we need larger pharmas that are making trillions of dollars to take on a humanitarian division. Maybe not to start them, but when they're there to take them forward. Because they are going to make millions of dollars. They just won't make hundreds of millions of dollars.
Yeah, but I think but to your point, I think the advocacy is going to be really important. Of how do you really push this ahead or make it a priority? Given that many of these subtypes we're talking about rare and small numbers, and it has to be prioritized. We can't get that far and have programs dropped. So yeah, all right. Well, yeah, I guess we have to end the session. But yeah,
Thank you so much.
Thank you everybody. Thanks to our speakers.